Let's do the walkthrough for the macromolecules lab. So the macromolecules lab, you start out, the walkthrough is right here. The lab handout, and I'm going to click on it and open the lab handout real quick so you know what you're looking at. So the lab handout, they usually are structured kind of like that. So the lab handout gives you background information. And the two things we want to talk about in this week is we're going to talk about the scientific method specifically, and then we're going to talk about macromolecules. So the first thing, we're going to talk a little bit about the scientific method, about how to do observations and formulating a hypothesis, about independent and dependent variables. And then we're going to talk about macromolecules, and I'm going to go over all of that in the lecture. Once you're done hearing about macromolecules, then we're going to get to the things that you're going to actually do. And what we're actually going to do is we're going to use 4 different reagents that will allow us to detect colorimetric whether there is a specific substance in an unknown. So, for example, we have 5 tubes here. Yes, there's distilled water in this one, glucose in that one, starch, ovalbumin, and corn oil. And you need to know that this is water, this is a sugar, a carbohydrate, this is a polymer, a sugar polymer. Ovalbumin, that's going to be your protein, and corn oil is going to be your lipid. So this is going to be the examples for the 4 different macromolecules you have. And with Benedict's turns orange when you have a sugar. If you have a little sugar, then Benedict's turns green. So those are the kinds of things that you're going to be looking at. And then so we've got that. We've got iodine that we're looking at. We've got burette, which tests for protein. And then this whole thing culminates into a bunch of unknowns that you need to discover, that you need to figure out what they are. So in these first pages, all you need to do is figure out whether they're positive and negative. And then where students usually get really iffy is when you get to this page and you're like, how am I supposed to figure that out? Don't sweat it, because in the quiz you have options. So let's go. So this was the handout. In the background information, I go over what macromolecules are, what these tests are, what a positive and what a negative test looks like, and then we're gonna look at some more about the tests and the positive and the negative. And then the macromolecules quiz asks you about the questions in the lecture. And then I'm going to click real quick on the handout quiz. So when you look at the macromolecule, when you look at the handout quiz, you'll find that it basically has the same images on it as your handout does, as your question set does, right? And it basically, you're putting the answers from the question set in there. And the part that I wanted to show you specifically is when you get to the unknowns, you're being asked for what's positive and negative. Here, you're being asked for what's positive and negative, but you're not having to guess 100% at what is what, because when you get, oh, I don't even ask you that. Oh, no, yes, I do ask you that. When you get to the questions, you have options so I'm giving you 4 options of what this thing may be, so that you're not, guessing out of the blue. When we do this in the lab, then we can actually smell too, and so you can have an idea of what something is based on that. So like I said, when you get to the handout and you're like, I don't know how to even figure this out, don't despair, because you can, in the quiz, it's not going to be as hard. And that's really it for the macromolecules. Oh, wait, one more thing, the labster. So the Labster, so for Labster, I want you to do lab safety, and then the introduction to food molecules, which is actually the same tests: Benedict's, iodine, Sudan 4, and burette. Watch out for the Sudan 4 a little bit. When you go online and you look at the Sudan 4 online, Sudan 4 comes in different preparations. The one that we have in our lab is a liquid. And actually, I think even the Labster activity may be different from the way that we do it. It very much depends whether it's a liquid or a powder. The other items that I want you to consider looking at this week when you click at the Labster simulation. So there's lab safety, there's an introductory lab. The introductory lab is probably going to be better for the enzyme lab, because we're going to be talking about acids in it. And then you've got the introduction to food molecules. If you want to go look at the scientific method already, be my guest. And enzyme kinetics, be my guest too. But like I said, these are your options. Just do 2 out of those. And like I said, anything you do extra is going to be bonus. And that's, I think, it for the macromolecules lab.